The messenger RNA has now completed its journey out of the nucleus and entered the cytoplasm. Unlike DNA, which remains safely protected inside the nucleus, this temporary copy carries the instructions to the cellular machinery responsible for building proteins. Ahead of us, the next stage of gene expression is about to begin. As we move along the messenger RNA, one particular three-letter sequence comes into focus. This start codon, August, acts as the universal signal that tells the cell exactly where protein synthesis should begin, ensuring every protein is assembled from the correct starting point. Approaching the start codon, the smaller ribosomal subunit docks onto the messenger RNA with remarkable precision. Rather than reading the entire molecule at once, it carefully positions itself at the correct location before protein production can proceed. Another ribosomal component now arrives to join the first. Together, these two subunits surround the messenger RNA, forming a stable platform where the genetic instructions can be translated into a chain of amino acids. The ribosome is now fully assembled and ready for action. This remarkable molecular machine does not create information itself. Instead, it carefully reads the instructions carried by messenger RNA and converts them into a completely different biological language, the language of proteins. Our attention now shifts to another essential participant in this process. Floating through the cytoplasm is transfer RNA, or tRNA, each carrying a single amino acid that will become part of the growing protein. Together, thousands of these molecules continually supply the building blocks needed for life. As we move closer, the lower end of the transfer RNA comes into view. This region contains an anticodon, a sequence of 3 RNA bases designed to recognize and pair with one specific codon on the messenger RNA, allowing the correct amino acid to be delivered. The transfer RNA pauses briefly above the messenger RNA as both sequences are checked for compatibility. Only when the anticodon matches the codon perfectly does the ribosome allow the molecule to enter, maintaining the remarkable accuracy of protein synthesis. The match is successful. This precise molecular recognition ensures that the amino acid carried by the transfer RNA corresponds exactly to the genetic instructions encoded within the messenger RNA, preventing mistakes before the protein continues to grow. Now another transfer RNA approaches, carrying the next amino acid in the sequence. Inside the ribosome, neighboring amino acids are brought close enough to interact, preparing for the chemical reaction that will permanently link them together. At the heart of the ribosome, a peptide bond forms between adjacent amino acids. One by one, these bonds transform individual building blocks into a continuous chain, gradually converting the genetic code into the primary structure of a protein. With one amino acid successfully added, the ribosome advances exactly one codon along the messenger RNA. This movement, known as translocation, shifts the reading frame forward while preserving the correct sequence of the growing protein. The transfer RNA that has already delivered its amino acid has completed its role. It leaves the ribosome to be reloaded with another amino acid elsewhere in the cell, while space opens for the next transfer RNA carrying the following instruction. As translation continues, the ribosome acts like an efficient assembly line. Amino acids are added one after another in the exact order dictated by the messenger RNA, allowing the emerging protein to extend steadily from the ribosome. The growing chain now extends beyond the ribosome as a newly synthesized polypeptide. At this stage, it remains a simple linear sequence of amino acids, but that sequence alone contains all the information needed for the protein to eventually fold into its functional three-dimensional structure. Eventually, the ribosome reaches a stop codon. Unlike other codons, this sequence does not recruit a transfer RNA. Instead, it signals that the complete protein has been synthesized and that translation should come to an orderly conclusion. With protein synthesis complete, specialized release factors trigger the final steps of termination. The finished polypeptide is released, and the ribosomal subunits separate from the messenger RNA, ready to participate in another round of translation. Freed from the ribosome, the newly synthesized polypeptide begins an entirely new phase of its life. Although its amino acid sequence is complete, it is not yet fully functional. The chain must now fold into its unique three-dimensional shape before it can perform its specific role inside the cell. Our journey now follows this newly formed protein as it leaves the ribosome behind. In the next chapter, we will explore how proteins fold, are modified, and are directed to their correct destinations, revealing how the genetic instructions encoded in DNA ultimately become the molecules that power every living cell.